Today's OCCPR webinar will be about NCI's Proteomic Data Commons. Dr. Chris Kinsinger, Program Director at OCCPR, along with Dr. Rajesh Thangadu, Project Manager at ESAC Inc., will talk about the program, the portal, and how to submit data. During this webinar, all attendees will be muted. If you have any questions during or after the presentation, please submit them through the webinar chat box. This webinar will be recorded and posted to the OCCPR website. We are glad you can join us.
Thank you for joining the NCI's Office of Cancer Clinical Proteomic Research webinar series. As a part of NCI's Cancer Continuum, OCCPR mission is to improve prevention, early detection, diagnosis, and treatment of cancer by enhancing the understanding of the underlying molecular mechanisms while advancing proteome and proteogenome science and technology development. Through the integration of genomics and transcriptomics with proteomics, also known as comprehensive proteogenomics, OCCPR's aim is to advance basic, translational, and clinical research through OCCPR-supported programs. This webinar series hopes to educate you to all the resources OCCPR and its researchers have to offer.
Okay, so once again, welcome to the first webinar in the OCCPR webinar series. Today we're talking about the Proteomic Data Commons. I'm Chris Kinsinger, and I'm joined by Rajesh Thangadu of ESAC. I'm a program director at the NCI's Office of Cancer Clinical Proteomic Research, part of the Center for Strategic Scientific Initiatives. So the Proteomic Data Commons, before I get into it, I want to acknowledge the many people who have worked on this project so far, led specifically by Rajesh and his team at ESAC, as with help from University of Washington, Mike McCoss, Paul Rudnick, also some help from Georgetown University, Nathan Edwards, as well as other folks at the NCI, and quite an esteemed array of people on our scientific advisory committee. And also I want to point out Manisha Ray from Seven Bridges Genomics. She's worked to connect this resource to the Cancer Genomics Cloud resource at Seven Bridges, which you'll see a little bit later. Okay, so what we're going to go over today is Proteomic Data Commons in the context of something called the Cancer Research Data Commons. And then I'll hand it over to Rajesh, who will just kind of show some general navigation of the PDC, and then go through some specific use cases. How would a cancer researcher use the commons? What's one way a cancer biologist would use it? And then if you're more of a bioinformaticist, we recognize there's different users and the way you're going to interact with the data looks a little different, so we'll go over that as well. And finally, it'll be an invitation and show you how to submit data to the PDC, and we'll end with time for questions. All right, so just some context. As I think about data repositories, I think there's kind of two types. So on the left-hand side, you have repositories that by data type, so it might have a repository full of genomic data or proteomic data or imaging data or clinical data or another type, and it serves that community very well. But you also might be doing an experiment that involves multiple types of data, and so then what might work better for you is a repository by program. So something like the Cancer Genome Atlas had a data coordinating center where you could go to get all of the data. The Clinical Proteomic Tumor Analysis Consortium also has a data coordinating center that has quite a few types of data stored there. And that's really helpful because you get the data for those experiments all kind of integrated together, and it's kind of a one-stop shop. But it falls short when maybe you're not part of that program. Maybe you have data that would be complementary to it that isn't able to go in to that data coordinating center repository. And so I think as we're getting to kind of the next generation of repositories, there's kind of this middle ground, something that goes beyond the repository of a single data type that aggregates data of different types and is integrative in nature, and something that also isn't specific to one program or one group of researchers, but is more transcendent across research communities. Another aspect is, a lot of us kind of got our training in one technology, and so that might lead us toward a repository by data type. But as you get into working in a disease area, then you're looking at data from a variety of technologies. And so I think the next generation of data resources is really going to have to address that, to be in the context of a disease, to provide access to many types of data, and more than that, not just to bring them together, but to really integrate them so that you can use them wisely to generate hypotheses and both get biological and clinical insights into the disease you're studying. So with that, I want to introduce the NCI Cancer Research Data Commons. So this is a work in progress that was launched really with the 2016 moonshot that came from the White House. And so one aspect of that was to generate a cancer data ecosystem. And so what you can see here, the core of it are these nodes, these cylinders here, that are really repositories by data type. But what you have around them is a data commons framework that unites the data within each node. And so the data commons framework has things like a common way to access the data, common authentication and security systems. It has a common indexing system so that all the files are indexed in a common way, and other common services that make the Cancer Research Data Commons more accessible and accomplishing those goals I set out in the previous slide. There's a number of portals, but we also have a number of application programming interfaces. It's all based in the cloud, and we have some cloud resources where you can do quite a bit of computing that can, just point to the buckets of each of these data nodes. Okay, and the Proteomic Data Commons is one node within the Cancer Research Data Commons. So we're going to talk about that a little bit more specifically now. So our goals for the Proteomic Data Commons are to connect proteomic data to other data types. As you'll see, we want to make the proteomic data more accessible to especially biologists and clinicians. We're moving to a cloud-based workspace model. And in addition to releasing the data, we'd also like to release workflows and software pipelines with the data so that users can do their own analyses on the data. And finally, we'd like to improve metadata annotation of the proteomic data sets. So let me just back up and talk a little bit about proteogenomic integration and what I mean by that. So a lot of this is derived from a program called Clinical Proteomic Tumor Analysis Consortium, where we're taking tissues from human tumors and characterizing them for genomics and for proteomics. We also get the histology images and the medical images, and all of those become available mostly through the Cancer Research Data Commons. So why would you want to do that? Why would you want to look at a tumor in so many different ways? Well, at the most fundamental level, you could use it to identify variant protein sequences corresponding to somatic mutations or germline mutations in the tumor, and see which of those variants at the genomic level actually get expressed at the protein level, and does that tell us anything interesting? We can look at relationships between things like copy number variation and protein expression, or gene expression and protein expression. And then ultimately we can kind of derive a new taxonomy, or not a new taxonomy, but kind of enhance how we understand a specific cancer by applying not just DNA data, not just RNA-seq, but looking at DNA, RNA, and protein, all these molecular features, which are all important to the cancer biology. So that's kind of the background on what we're trying to do with proteogenomic integration. So at this point, I'm going to hand it over to Rajesh Thangadu, who's going to go through the framework and how to navigate the Proteome Data Commons. Thanks.
Thanks, Chris. So I think that helped to understand where we stand about this proteomic data commons in the context of the science services data commons. So what you see on the screen right now is basically a schematic of how different components of the proteomic data commons. So we have a data portal, so where all of the data can be actually visualized or analyzed, browsed or downloaded. It does a lot of different functions. And also we have a workspace component where the users can actually submit their data and organize that. I will jump right into the demo. So here, like Chris mentioned on his first slide, I will run my demo through a series of use cases based on which we actually developed this resource. It's not just one resource we started developing and finding users, but rather with the help of our advisory committee, we try to identify different use cases so that the resource can be helpful to a lot of different kinds of users, not just the cancer biologists, but also cancer researchers and also the proteogenomic researchers. So I highlighted them here on the slide, but I will jump to live demo, so hopefully it goes well. All right. So great. This is how you access Proteomic Data Commons. You can simply type in pdc.esacking.com. So, and that's the URL for the Proteomic Data Commons. And right now we are in the development phase, what we call is a beta phase. But when we turn to production or the full production, it will turn to cancer.gov domain. All right, so this is the home page which provides you a lot of different, it summarizes all of the data within the Proteomic Data Commons. So, and it has a lot of different navigational elements. You can simply click on any of these primary sites. It's currently displaying how many number of cases are there. And also on the right, you see disease types. So here it's telling how many number of cases are there for each disease type. So by cases, I mean basically it has different terminologies at different resources. A case can be a subject or a participant or a donor or a patient. Different programs call it differently, but at the end of the day, this is the tissue provider source. All right. So I want to highlight, before I jump into actually how do you browse all of this data, I want to highlight that we just released the pediatric brain tumor atlas data last Monday. So that's the freshest data. So I will try to click on that pediatric brain tumors here. So once you click, you directly jump into the browse page with those applicable studies selected. So as you can go down a little bit, you'll see there are two studies displayed here, the pediatric brain cancer proteome and phosphoproteome. So let me clear the filter. You can just clear the filter here. And the chart at the top here is also interactive, so you can click and everything will be updated on the page, but you can clear that. And if you want more area on your screen, you can just simply collapse that. So you will see here, so there are, so in the center you see this table. This is actually representing all of the data that is there in the Proteomic Data Commons organized as studies. So there are 5 different tabs: studies, biospecimen, clinical, files, and genes. These are all interrelated. Here it's trying to tell a little story about, right now there are about 24 studies in the Proteomic Data Commons derived from, it includes about 1,887 biospecimens, which are derived from about 1,300 patients. And there are about this many number of files. And out of these 24 studies, a total of 13,000 close to 14,000 genes were identified. So as you, on the left, you see a lot of different filters. So we call it as, we try to group them like general, biospecimen, and clinical, and so on, so that it helps you to understand those filters instead of just digging through a lot of different ways you can filter the data. For example, if you want to just look at breast cancer data, you can just click that. And then if you want to look at a certain demography, for example, you just want to look at Black African American, you can just click that. So as you can see here, automatically all of these tabs are updated to reflect your selections. So right now there are 4 studies that are displayed here. And if you, for example, if you click on the clinical data, it actually shows only the cases from those 4 studies which have the clinical parameters that you selected. So for example, the black or African American, that's the race you selected here. So that will allow you to quickly navigate through all of the studies. It's not just one single study, but all the studies. All right, so let me clear the filter. And if you want to learn more about a particular study, you can simply click on any of the studies. So it will open up a study summary page, what we call. It provides much more information about that particular study without need for downloading any single document. So here, there is some quick summary which is all self-explanatory. There is a good description about the study. And also the protocol tab describes the actual protocol used to generate the data for this study. Similarly, the clinical information, the same tab that you saw earlier, but this is in the context of this particular study. And also you have the biospecimens. And finally, the workflow. So the the Proteomic Data Commons has a common data analysis pipeline through which all of the data will run through. I will talk about it a little bit later. But the idea here is, so we will collect all of the metadata information about how exactly the study has been analyzed. That information is available, the tools, their versions, and so on. And finally, if applicable, there is a data use agreement also displayed. Depending on the programs, they may have different data use agreements. So we have that information available here. So there is a way to get into the individual files. You can click any of these links to jump into those links. And when there is a publication available, it will be updated here. Right now, this study is not published yet. And there is a way to look at the heat maps. I will come to that in a bit. All right, so that's about the general navigation. So as you can see, all of this can be done only if you have rich metadata and also a robust data model. So you can go to the help to understand more about that. So there is a data model and dictionary that is guiding all of that's driving all of the PDC resource. You can just click that if you want to learn more about the data model and how do we organize. So if you click on the explore data model, this is all of the different ways, all the entities within the Proteomic Data Commons are connected and how we organize the data. So I don't want to go deep into that, but I'm just pointing that, so if you have time, you can explore that further. And also the data dictionary, basically what it means is for every element that we describe, we try to follow the standards and terminologies that are available in the literature. For example, if you go to the clinical, if I click diagnosis, it's basically telling what are the different information that PDC intends to collect. This is all not required, but we encourage the data submitters to submit as much information as possible. So it provides a description of each of those elements and also their connection to the cancer data standards repository, as you can see here. All right, so let me go back to the home page. So I think I tried to explain how a cancer researcher can browse the portal, but if a cancer biologist comes to the data portal, he doesn't really want to spend a lot of time here, but they just want to look at, is my gene present in this data that is available on the Proteomic Data Commons? So what can they do? So like I mentioned earlier, there is a common data analysis pipeline that is run on all of the data within the Proteomic Data Commons. So what that basically means is, as a part of this common data analysis pipeline, people who are familiar with the CPTAG data portal, they can simply, they are aware of this CDAP, common data analysis pipeline, but here the protein assembly reports are generated at the level of genes. So to make things simpler, the one reason is to doing this rolling up to the genes instead of proteins is that, so sometimes it enforces some inaccuracies in how we assign the abundances to minor protein isoforms. And also it's pretty common in the proteogenomic era for a lot of genomic biologists, they will come to these kind of resources with a gene rather than a protein. So for that reason, the CDAP outputs are rolled up to the gene level. That's why we have the genes here. So if you are interested in a particular gene, you may simply enter the gene, search for the gene, and it will automatically pop up the particular gene, and you can search quickly and go to the full summary to understand the gene. So the other way you would do is, you can simply go to the genes tab. As soon as you go, So there is a way to search multiple genes. Sometimes you are interested in not one gene, but a group of genes which are from a pathway that you are interested in. So either you can simply start entering them with a space here, or we also have provided a quick and easy way of looking at sets of genes that are quite popular in the cancer research area. For example, from this list, I can select ovarian cancer protective tumor suppressor genes in ovarian cancer. So it populates all of the genes and automatically verifies that all the gene symbols are valid. So you submit the query, and as you can see, right now you are just seeing those genes that you are filtered based on. So, and this view actually provides a quick summary of how many studies each of these genes are actually found in within the PDC. And if you want to understand more about the gene, you can just click on any of this gene and click full summary, and it provides a lot of different information. So there are not a lot of visualization on this page, which we are currently working on, but the idea about this gene summary is what is the status of this particular gene across all the data sets within the PDC. So on the left, you again have a lot of different filters. For example, you don't want to look at all the studies you are interested in, only breast cancer. For example, you can just select breast, and it will update to show only the breast cancer studies and all the aliquots or the actual samples that are used, and also the log-to ratios or the abundances. All right. That's one way. That's one thing you can do if you are looking for individual genes. Sometimes you just want to actually explore the expression data. What can you do? So you can click on a particular study if you are interested in. For example, here I'm looking at the kidney cancer data set, and you can click the Heatmaps. There is a lot of data, so it may take a little bit of time to load. That's not too bad. All right, so all of the expression of the protein abundance data for this particular study has been loaded as a heat map, and it's available. So we are using a tool from the Broad Institute that's Morpheus, pretty popular. The heat maps will actually provide a lot of interesting information instead of, without actually need for the download. So here you have about almost 10,000 genes. So you probably want to just look at only the highly variable genes. For example, let me create a quick column based on the median average distribution. So the idea is, can I filter? So I added a MAD, median average distribution column for the genes, and then I will filter them. So what I will do is I will select this particular column, and I will try to select the top 50 genes which are highly variable within this particular data set. All right, so as you can see on the left, so right now it's not showing all the 9,000, but rather 50 out of the 9,000. All right, so then you can go ahead, and if you want to do some kind of clustering, you can start to cluster. And you might have noticed that at the top, along with the samples, you have a lot of clinical information annotated for each of the samples. So if you may want to simply cluster by grade, and then run your hierarchical clustering. So the idea here is basically it will allow you to do a lot of analysis right on the interface without need for download of any of this data. Because the clustering usually it takes a little bit of time, let it run, and then here, yeah, it's there, and as you can see, it simply nicely clustered things together, and you can actually generate hypotheses right here. And you can either save these images, save this data set, or save this session, and come back later and continue your analysis. So these are a lot of these features are the part of the Morpheus. But the idea why I'm trying to emphasize here is that without downloading the data, you can do a lot of analysis here itself. All right. So let me move to the other use cases. If you are a proteogenomic researcher or a bioinformatician, what you can do. So that's what I'm trying to see here, talk in this particular use case. Let me go back to the screen. So you probably have noticed, but I did not talk about it. So on the clinical tab, when you click here, so we have a explicit column that's named as external data resource. Basically, what it is trying to say is, for the cases within the PDC, they have some complementary information elsewhere. So here, this is a data set for the Kids First, the pediatric brain cancer data set that I talked about earlier. So we have an explicit link. So the mouse over actually tells, go to the genomic resource. If I click on that link for this particular case, it will take me to the Kids First data portal, where they will provide more information about that particular case. Similarly, if you are interested in breast cancer, so this is related to the CP tag. So here you have a link to the genomic resource, that is the genomic data commons. You can click on that. It will go to the GDC, and you can learn more about what information is available for that particular case or a patient on the genomic data commons resource. So we also have a link for the cancer imaging atlas. So you can just click on that. It will jump to the cancer imaging atlas. So where you have the imaging information for this particular study. So there are direct links from here. That's the first step towards knowing what complementary information is available for this study or for the patients at a different resource. All right. So now I want to talk about actual, as in a lot of proteogenomic experts, they want to actually analyze this data together, all this complementary information, how we can actually analyze the data. Let me go to the slide. Let me move here. So this is a quick slide which describes the actual integration that we are working within that CRDC framework. All right, so I don't want to spend too much time here, but just to give you an idea, so these green boxes out here, they are like different nodes, PDC or genomic data commons or the imaging data commons. And on the right, you have the cloud resources. Probably some of you are already aware of some of these cloud resources, something like the 7 Bridges Genomics, Broad Institute's FireCloud, and also ISB's CGC. And also, a lot of institutes probably have their own cloud resources or organizations, so they can always connect to the PDC or GDC or other resources through a framework of services that's called the DCF that has been established. So we recently piloted with the 7 Bridges CGC. So in very basic terms, what the user can do is you come to the proteomic data portal, identify the files that you want to analyze, and just simply download that manifest and take that file. It's not actually downloading the files, it's the list of files you want to analyze. It's a simple text file, and you take it to the Cancer Genomics Cloud. That's here, I gave the URL here, and you simply upload that file, and the system will automatically pull all the files from PDC. And on the CGC, you have a lot of different tools available to analyze the proteomic data. At the same time, I will show some screenshots here. They have some introductory tutorial how to do this. So if you go to the CGC, 7 Bridges CGC, you will see some information there. So here is a screenshot about how you select all the files and download the manifest. And you go to their interface and load the manifest, and you have all the files as you can see, the MGML, some other things. And then the previous examples, use case, I talked about the clinical tab. So you have the clinical information in the manifest file. We actually, when you download, you would notice that the GDC case IDs are there. So the CGC platform automatically pulls this information from the PDC manifest and pulls in all of the genomic information from GDC. So what the user is actually doing is simply providing a manifest, nothing else, but our interoperability approach is actually trying to bring in all the things together, and also you have a set of tools that are available for you to analyze the data. So that's where we are. It's a work in progress, but I mean, we are happy to provide more information if anyone is interested to do this. And finally, we do have a lot of APIs. So whatever I showed on the interface, the user interface, you can do all of those things programmatically through the APIs. If you go to the, let me go back to the Proteomic Data Portal, and here on the help, you can click on the API documentation. So we have a pretty good, actually a pretty extensive documentation available there. You just read through this, and we have a public Swagger. What this means is you can click on that, and it has a lot of different, it lists all the different APIs that are available. So you can try these APIs right here. I don't go to too many details here. So you can try those APIs right there. And also, we also provide some actual examples how to use the APIs in a Python script. For example, if you click an example here, here is how we wrap all these APIs in a simple Python script to generate a heat map. So basically, this particular Python script, what it is doing is querying the PDC programmatically and getting all of the expression data for a given study and drawing a clustergram. So what this basically means is you can do all of these things programmatically as well. And finally, one last use case I have is, I talked a lot about how a user would use the PDC resource, but there will be users who want to actually submit data. The submitting data is not necessarily an endpoint for PDC, but you can actually see your data sets alongside all of the other very important data sets that are available there, so that it will allow you to slice and dice your data in comparison with other CPTAC or pediatric brain cancer or Apollo data sets that will come in future. So please just write to us, nci.pdc.help@esacking.com. That's our help email. But how it works is we will set up a program. If you are interested in submitting data, we will set up a program for you, and you would have to register to PDC, and then we will provide more details. I think at a future time we will have a separate session for how to submit data to PDC. But for now, so you can go here. Usually, you do not need to log into PDC. You see that login button at the top, but you would need to log in only if you want to submit data. So if you click submit data, it will ask you to log in. So the good thing here is either if you have a Google login or NIH or NIH ERA Commons accounts, you can simply use them to log into the portal. So you don't need to remember an extra password. If not, you can click here and you can create a separate PDC specific login. So once you are registered, so it will guide you through some of the other screens that I cannot show here, but there are some test programs we created. If anyone is interested, they can just simply join those test programs and explore the submission data portal. Or if you have some data sets you really want to submit, just let us know. We will set up a program for you, and we will also give you an introduction how to submit. I think I tried to cover different use cases, what you can do with PDC. There may be other things that I did not mention here, but feel free to go to our portal and try to spend as much as time you can, and let us know what you like about that, and if there are places that you think we can improve. We always welcome suggestions, and the user feedback is the one that's guiding development of this resource. Thank you.
Okay, thank you, Rajesh. So we have a few questions. I would also invite participants to go ahead, and this is a great time to type in any questions you might have about the presentation. So first question is, what data sets are available in the Proteomic Data Commons? And I think I can answer that. So most of the data have come from the CPTAC program, so we have data sets. They're all about 100 tumors or so in size, and we have that for breast cancer, ovarian cancer, colorectal cancer, kidney cancer, endometrial cancer, and lung adenocarcinoma. And then just yesterday we released a data set from pediatric brain cancer. And that actually came from the Children's Hospital of Philadelphia. So that's, and then there's one other, what's the one other data set? That's the SWATH?
SWATH is from Ebersol Group.
Yeah, from the Ebersol Group, we have a SWATH data set. All the other CPTAC data sets are either iTrack or tandem mass tag kind of proteomic profiling data sets. Okay, next question is, can you just show again, where do I go to link to corresponding genomic or imaging data?
So you go to the home page, you click on browse, and then you click on the clinical, and there you have the external data resource.
Okay.
And you can select all the files here, all of this information, not the files, just all the information, and click on download the clinical manifest. That manifest will have the genomic data commons IDs. So I would recommend to filter your data set to the specific study so that it will give you only that particular study.
Okay, great. This one is about data submission. Will my data go public as soon as I submit them?
I can take that. So there is a harmonization process. So we do have some submission requirements. There are not I showed you the data dictionary, so there are a lot of terms that we would encourage the user to submit. But we do have some minimal requirements about what metadata we take. So once you have that, there will be a few weeks we need to make sure all of the data is accurate, and then we will run the common data analysis pipeline and release the data.
But it's not going to be public until you decide to make it public. Yeah. That was the best.
Yeah. Yeah. So yeah, I mean, again, so you have to actually tell us when you want the data to be released. So once we confirm that the data is ready for release, that comes back to the submitters when they want to actually release the data.
Okay. All right, this one, you mentioned registration towards the end of the presentation. Do I have to register to browse the data? And if not, for what activities do I need to register?
Like I mentioned, the registration is needed only for submitting data. But we put that login information because we are anticipating some kind of controlled access data in future. So if there is any controlled access data or protected information within PDC, then you would need to have authorization to access the data. At that time, the user would need to log in. But at this time, so you do not need to register if you just want to browse the data.
Okay. Are there similar diseases, or are there similar data sets for metabolic diseases? So at this time, we don't have any metabolomic data sets. You could certainly submit metabolomic data sets. And I think they would work pretty well, but we don't have, a combinated analysis pipeline set up to analyze metabolic data. So the short answer is no, we don't have any metabolomic data right now, but that's something that we're looking to expand into in the future. Okay, can you please show again where to find the Python code to generate heat maps from genetic expression data?
You can go to the help, and there is API documentation. And if you go a little bit further down, we have this GraphQL and Python example, and we have a link out there. We have an example right there below that section, and also you can click this link that will take you to the actual Python code.
Okay, great. It's great that there's synergy between the PDC and GDC. Are there plans to integrate interoperability with other data commons and or clinical data? So the answer is yes. And mostly we're working within the context of the Cancer Research Data Commons. But one connection we that'll probably be next is the Cancer Imaging Archive, which is slightly outside of the Cancer Research Data Commons, but the Cancer Imaging Archive, TCIA, has a number of CPTAC images, histology, and MRIs, CTs, PET scans, those types of images. And so we do plan to put links to those images in the PDC in the near term. So there's another question related to this. Is there any link to Uniprot, ClinVar, or other external databases?
Yeah, at this time we do not, but yeah, we intend to have those kind of connections in future.
Right. So, yeah, but thank you for asking that question. There are other interoperability connections that are in the works as new data nodes come online in the Cancer Research Data Commons. We're working pretty closely with the PDC to make sure that we're going to be interoperable with those. And a lot of it happens through the cloud resources where you can do the cloud computing and the Data Commons framework, which have those core services for all of the nodes. What is the long-term plan for current CPTAC data portal at Georgetown? So that's a good question. As I mentioned at the beginning, CPTAC has had a data coordinating center that's been run out of a website hosted by Georgetown University. And mainly people go there to download the data. And so what we're looking to do is kind of merge that data portal with the PDC, and the PDC would kind of be the next generation data portal for moving forward. But that hasn't happened yet. Right now they coexist, and that'll probably continue for at least another year. Okay, well, that brings us to the end of the questions. Thank you so much for everyone for joining today. And again, the webinar has been recorded, and we're going to make it available on the website proteomics.cancer.gov. And finally, I'd like to hand it over to our production specialist, Anna Roberts-Pilgrim, for some closing remarks.
Thank you for joining.
Thank you for joining the Office of Cancer Clinical Proteomics Research webinar series. Please visit our online resources included here and take a minute to answer our online survey. The Office of Cancer Clinical Proteomic Research is part of the U.S. Department of Health and Human Services at the National Institutes of Health, National Cancer Institute. For more information about OCCPR, please visit www.proteomics.cancer.gov. For general information about cancer or clinical trials, please call 1-800-4-CANCER.